Welcome. In this session, firstly, I'm going to discuss about packaging of DNA helix, and then later I'm going to discuss about the search for the genetic material. So let's begin this session with packaging of DNA helix. Right now, when scientists have taken an electron microscope picture of an eukaryotic nucleus, electron microscope picture of an eukaryotic nucleus. So what they have seen inside an eukaryotic nucleus is some bead-like structures they have seen. So this is how they have seen some bead-like structures. Right, and these bead-like structures are linked by a thread-like structure Okay, so these are beads and this is a string. So they called it beads on string like appearance. So they called beads on string like appearance. So what actually these beads on string like, so these are nothing but chromatin material. So it's a chromatin, and this chromatin is composed of DNA plus histone proteins. So it is composed of DNA and histone proteins. Later I'm going to discuss in detail so what these histone proteins and what they are made up of and so right. So when scientists have taken the electron microscope picture of an eukaryotic nucleus, so they have seen the chromatin as beads on string-like appearance. Right here, especially I'll be talking in reference to human cell. Okay, so here in human cell, I'm going to calculate the length of the chromatin material. Okay, so here let's calculate length of chromatin material. Okay, so for this to calculate this, we need to know the diploid content of human cell, which is 6.6 into 10 to the power of 9 base pairs. So in earlier sessions I mentioned 3.3 into 10 to the power of 9 base pairs, which is okay, haploid content. So here it is the diploid content which I mentioned here. And next we need to know the distance between two base pairs. So the distance between two base pairs is 0.34 nanometers. Right, now I'm going to calculate the total length of the chromatin material in meter. So for this I need to take 6.6 into 10 to the power of 9 base pairs into the distance between the two base pairs is 0.34 nanometers into 10 to the power of 9 meter. Then I'm going to get a value 2.244 meter long. Okay, so if I'm going to put all that chromatin material, right, and I'm going to get a total length of that as 2.244 meter long. Now, when we see a typical nucleus, okay, length, when we see it, then it has only 10 to the power of minus 6 meter. So it has only 10 to the power of minus 6 meter. Now the question arises, how this 2.2 meter long chromatin material is going to present inside a nucleus that is only 10 to the power of minus 6 meter. So that is what the packaging of DNA helix. So the DNA helix is packaged in such a way that fits inside, okay, a nucleus that is only 10 to the power of minus 6 meter long, right? So let's see in detail about this, and before I discuss how this packaging is seen in an eukaryotic cell, so here I'm going to give a brief note on how we see in case of a prokaryotic cell. So in prokaryotic cell, so here I'm going to mention this in E. coli bacterium. Okay, so here E. coli bacterium, and in this E. coli bacterium the DNA is not scattered throughout the cytoplasm. Okay, and we know that prokaryotes like E. coli, they don't have nuclear membrane. Of course, none of the prokaryotic cells have nuclear membrane, and here we see the DNA will be seen with large loops, and okay, so this is how you will be seeing the DNA in prokaryotic cells. So this is DNA, and this DNA is surrounded by large number of proteins, and these proteins are not histones. So earlier I have mentioned DNA plus histone proteins that make up the chromatin, but here we don't see in that way. Okay, so here these are just proteins, not of histone, right? And now this DNA, we know that it is negatively charged. So whether it is prokaryotic or eukaryotic, DNA will be always, right, negatively charged. And in this case, these proteins are positively charged, right? Now the DNA along with that positively charged proteins together, it constitutes something called nucleoid. So it's called nucleoid, a nucleus like, okay, structure that we see in case of prokaryotic cells. So here there is less organization, okay, in case of prokaryotic cells, but when coming to an eukaryotic cell, right, so histone proteins are present along with the DNA, and the packaging is seen in a very orderly manner, okay, that we don't see in case of prokaryotic cell. So this is how we see in case of prokaryotic cell, the DNA which is surrounded by proteins which are positively charged, right? Now let's see in case of eukaryotic cell, right? So in an eukaryotic cell, here I will be mostly referring human cell, right? And I told you earlier that we have seen some bead like structures when we have taken the electron microscope picture of an eukaryotic nucleus, right? And that bead actually is a structure called nucleosome. Okay, so such things we don't see in case of prokaryotic cells. We are talking especially in case of an eukaryotic cell, right? And that string what we see here is actually the DNA, right? And this nucleosome is composed of histones. So it is composed of histone. So histone proteins will make up a nucleosome, right? Here, let's see, each nucleosome consists of, okay, 8 histone proteins. So each nucleosome consists of 8 histones plus a DNA helix with approximately 200 base pair length. So around this nucleosome, the DNA that coils will be having 200 base pairs. Okay, so that approximately I have given here. So 8 histone proteins, okay, plus the DNA helix with approximately 200 base pairs length will make up one nucleosome. So let's see what are all those, okay, 8 types of, okay, or I'll say 8 histones, 8 histone proteins. What are all? So they are H2A, then H2B, H3 and H4. So 1, 2, 3, 4. Okay, so these are 4 different types of histones, but each one is present in 2 numbers. So this is present in 2 numbers, this is also 2, also 2 and also 2. Total we have 8 histones proteins. So that's why, okay, it's called a octamer. So as it is made up of 8 histone proteins, it is called histone octamer. It's called histone octamer, right? So let's see. So this is H2A, then H2B, then H3 and H4. Okay, and they are actually 2 in number, so just I'm mentioning here. Okay, so total, I mean each 2 in number, total we have 8. So that's why it's called histone octamer. It's called histone octamer. So that made up of 8 molecules that total will form as histone octamer, and around this, so this is H2A, H2B, H3 and H4. 3, H4, H2B, H2A. I'll just mention, okay. And around this, you will see the DNA coiling in this way, right? So this is the DNA molecule, right? And I told you the number of base pairs that are present in the DNA helix, which is seen coiled around the histone octamer, will be having 200 base pairs. Okay, and outside this, of course, this together, now this DNA and this histone octamer all together will form the nucleosome. So it's that nucleosome what we see here. Okay, right. And outside this nucleosome we find another histone, okay, protein, and this is H1 histone. This is H1 histone, which is present outside the nucleosome. So the nucleosome is composed of, okay, 8 histones called histone octamer plus the DNA that has 200 base pairs. And these histones are made up of basic amino acid residues. So we know already that a protein is composed of, okay, amino acids that are arranged, right, in a particular order, right. And these histones are made up of basic amino acid residues that have positive charge on their side chains. So they are going to have positive charge on their side chains. So histones are made up of basic amino acid residues that have positive charge on their side chains. Now, what kind of basic amino acid residues found in those histones are actually lysines and arginines. So these basic amino acid residues are mainly, of course there are many other, but okay, mainly have lysine. So lysines are amino acids and also arginines. Okay, so these two are amino acids. Okay, so mainly have lysines and arginines. They are basic amino acid residues and they will be having positive charge on their side chain. So with this we understood that this histone octamer is positively charged. So it is positively charged. So the positive charge is due to presence of those basic amino acid residues that have the positive charge on their side chains, right? And we already know that DNA is negatively charged. So it is negatively charged. So this negatively charged DNA will be wrapping around positively charged histone octamer, right. So let's see how it looks. This beads on string like, if I'm showing this one with the histones, so I'm going to put these 4 histones together as one circle. So let's see. So this is one histone octamer, this is another histone octamer, and this is another histone octamer. And now we will be seeing the DNA is seen wrapping around something like this, right. And outside this we have H1 histone. So this is H1, H1 and H1 histone, and this is nucleosome. Okay, and this is again the nucleosome, right. So now we understood that what actually those beads and strings, this is what the nucleosome which appeared like bead, and the DNA which actually joining, okay, one histone with the other look like string. So this small stretch of DNA that is present between two nucleosomes is actually called the linker DNA. Okay, so this will be in the double helix structure only. It is not linear. Actually, even though just I drawn a line, it is in the double helix model only, that will be seen coiling around, okay, each histone. So this is histone octamer. So this is also histone octamer. Right. I hope this is clear. We understood what actually those beads on string like, right? So this nucleosome is the one that appeared like bead, and that DNA that appeared like a string. And such, okay, arrangement is not seen in case of prokaryotic cells. So the DNA is seen with associated with some proteins, but we don't see arrangement in this way. Now, okay, so this chromatin is going to coil upon itself and form chromosomes during cell division. So that's we will see. Right. So this chromatin will be coiling and result in the formation of chromatin fibers, and that chromatin fibers will finally result in the formation of chromosomes, right? So here they will be actually coiling in a spiral fashion. So here there is right, a nucleosome. This is one. Okay, so you see, okay, six histones here, and you can see here the DNA coiling around it. Right. Okay, so you will be seeing something like this. So this is a histone octamer, and this is the DNA that coils around. So actually when you see, it is seen like this. So this is the portion which I have actually shown here. Okay, so this the dotted one which I am showing here, that has 1 2 3 4 5 6 histones, right? And it will be seen all, okay, in this way here, and this forms something called a 30 nanometer fiber. It forms a 30 nanometer fiber. When we see in case of the nucleosome, so the nucleosome, okay, and with its DNA coiled around, and this nucleosome usually have 11 nanometers. So it has nucleosome, this has 11 nanometers. Okay, so the length will be 11 nanometers, and you can see this is one nucleosome, and they're all in spiral fashion, they are arranged. Okay, so this is how we will be seeing, right? And this is what I call a 30 nanometer fiber, is called 30 nanometer fiber. And this 30 nanometer fiber is going to coil further, right? So from here, so the coiling is much more. So we'll be seeing something like this, and this results okay, coiling and coiling and coiling, of course this results in the formation the next one I'll mention as 300 nanometers, and after that it finally results in the formation of one chromatid, so that will be having 700 nanometers. So here you will see something coiling, just I'm showing here something like this. Okay, so it's all coiled in a proper manner. Okay, so that they can even uncoil very easily. So this is how we see. Okay, and this one is a centromere. Okay, right, and this the chromatin fiber will be having 700 nanometers. So we know this is one chromatid and this is another chromatid. So both together it results in formation of 1400 nanometers. So this is okay 700 nanometers and this is 700 nanometers. Okay, uh, right. So here it's very difficult to show you in a three-dimensional way, right? So you can search on Google, just type in the Google packaging of DNA helix, and in one picture itself, of course there are lots of pictures. In one picture itself they will show very detailedly right from the nucleosome to the formation of chromosome, right? So what this chromosome it consists of, if you start pulling it from one end, okay, so you will be getting some nanometer fiber, chromatin fiber, and that goes up to 300 nanometer fiber, and further uncoiling and uncoiling you will get 30 nanometer, okay, chromatin fiber. And if you uncoil that one also, you will be seeing, okay, a chromatin that will be striped with the histones coiled around by DNA, right? So this is how we see the packaging of DNA helix, and is seen inside an eukaryotic nucleus, right? And here the packaging of chromatin at higher levels, packaging of chromatin at higher levels. Of course, at lower levels, we have seen the histone is required. So histone proteins will help in packaging the DNA at lower levels, but at higher levels, okay, of packaging the chromatin material, so it requires non chromosomal, non-histone chromosomal proteins. So it requires non-histone chromosomal proteins. So this in short I call NHC, right? So non-histone chromosomal proteins are required, okay, to pack the chromatin at higher levels, right? Now I hope it's clear. Now if I stain the chromosome, so if I'm staining the chromosome, I see that some regions of chromosome are lightly stained and some are darkly stained. Say suppose this region is showing dark stain, okay, so something like this, right? So these are darkly stained regions. So in case of a chromosome, and these are lightly stained regions, right? So when I applied stain, so we can see that the chromosome shows some darkly stained regions and lightly stained regions. So what about these darkly stained regions? So here, in case of darkly stained regions, we see that there is a tight packing of DNA. So that's, I say, densely packed chromatin. So in the region where we see dark stained, we see that the chromatin is densely packed, so tightly packed. And in case of lightly stained regions, we see it's loosely packed chromatin, right? Okay, so let's see an example. In this case, take a thread and then dip it in a color, whatever the color you like. Okay, so say suppose you have dipped in the blue color, and then after drying up, take that thread and start, coiling around a pen, whatever. Now where you see more coilings, there you will see, okay, thick color of that blue. And if you put, okay, less coils, then that will be lightly seen. Okay, so where you put more coils of the thread, the darker will be the blue color, and where you put, okay, less coils of the thread, you will see light color of that blue color. Okay, the same thing here. Wherever we see on the chromosome the dark regions, it indicates that there the chromatin is packed, okay, tightly, and in this case it is loosely packed regions. So here, why it is in this way? So these densely packed chromatin, or what I call it as heterochromatin, I call it heterochromatin. And this one, which is lightly stained, where we find loosely packed chromatin, is called euchromatin. Okay, and it is in the heterochromatin where we see there is no transcription taking place. So this heterochromatin are the regions where transcriptionally there is no transcription taking place. So I say this heterochromatin is transcriptionally inactive. Is called transcriptionally inactive, whereas euchromatin is a region that I call it transcriptionally active, right? So what actually we mean by transcription? So transcription is a process of formation of RNA from one of the strands of DNA. So here, formation of RNA from one of the strands of DNA, right? How it takes place and what are all the mechanisms, we have a detailed topic about this in the coming sessions about the transcription. There I'm going to tell you in detail that how the RNA is going to form from the DNA. So formation of RNA from the DNA is what we call transcription. And for this transcription to take place, the DNA should not be in packed. So if it is packed, it cannot perform the process of transcription. So the DNA should be, okay, uncoiled state. It should be in uncoiled state so as to perform this transcription. So that is the reason, okay, why, okay, some regions of a chromosome, it means that here as it lightly stained, the chromatin is not tightly packed, and where the transcription is taking place actively. And in these regions, that is in heterochromatic regions, transcription is not at all taking place. So hence, okay, right, so they appear dark stained. right? And I hope it's clear. So that's all about the packaging of DNA helix. Next, we will move on to the search for genetic material. The search for genetic material. Right. Now, we know that a cell is mainly composed of proteins, lipids, right, carbohydrates, and then nucleic acids. Okay, so here in earlier days, say suppose before 1900, okay, no one knows what actually is being passed from, okay, parents to offspring. Is it the proteins are passing from parents to offspring, or carbohydrates, or the lipids, okay, or the nucleic acids? So no one knows out of these proteins, lipids, carbohydrates, and nucleic acids which one is genetic material. Which one is genetic material? Of course, we already have understood that it is DNA, as I have mentioned several times in previous sessions, but I'm talking in, okay, 1900s. So I mean, before 1900, even before 1926, no one knows actually what is the genetic material. So whether protein is acting as genetic material, or lipids, or carbohydrates, or nucleic acids. And the search was started, okay, in 1926 with the experiments of Frederick Griffith. Right, so let's see, in 1928, Frederick Griffith was doing some experiments on a bacterium. Okay, so he was in 1928, Frederick Griffith was doing some experiments on a bacterium called Streptococcus pneumoniae. Right, so this bacterium causes a disease called pneumonia. So this one causes pneumonia. So if pneumonia is a problem with the lungs, right, and the person will not be able to get enough oxygen due to this condition, and the person dies due to lack of oxygen, right? So here in 1928, Frederick Griffith, he was doing experiments, some experiments on this bacterium in the laboratory, and we know that these bacteria can be grown on a culture medium. So usually they will be growing these bacteria on petri dishes. So I hope we know petri dishes that we see in the biology lab, and this will be filled with nutrient medium like agar. Okay, so say this is agar nutrient medium. On that you put some bacteria, just one or two, and they start multiplying on it and form into colonies. So in a short period of time you will see so many, right? So they multiply and form so many. Okay, so these are called colonies of bacteria. So initially you inoculate it with a small number, but later you will see lot many growing in that location, right? So while working on this bacterium, Streptococcus pneumoniae, he found two strains of this bacterium. Okay, so pneumoniae, right? He found two strains of this bacterium. So here one of the strain is producing smooth colonies, is producing smooth colonies, and another strain is producing, okay, rough colonies. So when seen under microscope, some colonies, okay, are shiny. So it is due to presence of a mucus coat in this bacteria. Okay, so they produce smooth colonies because they have a mucus coat. So this mucus coat is a polysaccharide. A mucus coat is a polysaccharide. And in case of this other colony, so that appears to be rough under the microscope, means they are not shiny like this. So hence he mentioned rough colonies, and in this case they don't have mucus coat. They don't have mucus coat. Okay, uh, that's I'll just mention. So this is the bacterium, and this bacterium is covered with a mucus coat. So this is the mucus coat. And in case of the rough colonies, we don't find any mucus coat. Okay, so that's what he mentioned this as S strain. So he mentioned as S strain, whereas these rough colony varieties, he mentioned it as R strain. Right. So when Griffith was doing experiments on this pneumonia, streptococcus and pneumonia, he found two strains. So one strain, they are, okay, showing smooth colonies due to presence of that mucus coat, and the other variety, okay, are not showing that smoothness due to absence of that mucus coat. And the smooth strains, okay, the smooth colonies he mentioned as S strain, and the rough ones as R strain. So I hope you understood that the S here means smooth and here R means, okay, rough. Now what he did is he took S strain, okay, streptococcus pneumoniae bacteria, and he injected into mice. Right, I hope this is clear. So what he did, he took S strain, and then, okay, he injected into mice, and what he observed is that mice died later, and they died due to pneumonia. So here mice, okay, so they died due to development of pneumonia. Okay, and with this he concluded that this S strain is virulent, which means it is harmful or disease causing. So that's, I'll say, pathogenic, means disease causing. Right, so S strain is virulent or pathogenic because it caused the development of pneumonia and resulted in the death of mice. Then he took R strain and then injected into mice. Now when he injected this R strain into the mice, so mice were alive, they did not die, okay, and they did not develop pneumonia also. Of course, due to non-development of pneumonia, all these mice were alive, okay. And with this he concluded that the R strain is non-virulent or non-pathogenic, means it does not cause disease. right? So when he injected the S strain into the mice, mice died, okay, due to pneumonia, and he concluded that S strain is pathogenic, means that variety can cause disease, and the S strain is virulent. And R strain, when he injected, okay, into mice, mice did not develop any symptoms of pneumonia, and they were completely healthy and they are alive. And with that, he concluded the R strain are non-virulent or non-pathogenic, right? So now later, okay, so what Griffith did is he has taken heat killed S strain bacteria. Okay, so he took S strain, but they are heat killed, means by exposing them to heat, he killed this S strain bacteria. Okay, now then he injected, so after heat killing them, he injected into mice. Now what he observed is that mice were alive, they did not die, okay, because they did not develop pneumonia, right? Because he killed them completely, right, by using heat. So S strain bacteria, okay, heat killing, and then injected into the mice, and the mice were alive. Now in another experiment, what Griffith did is he has taken again S strain bacteria, and he heat killed them. Okay, so he killed them by exposing to heat, and then what he did is he mixed this S strain, that is heat killed, with the R strain, and this is live, right? And now this mixture he injected into mice. Okay, so this mixture he injected into mice, and what he observed that mice died. Okay, so mice, so they are dead. Now he took the blood of these mice, so the blood sample of these mice have shown the presence of live S strain. Okay, so the blood sample of these mice have shown the presence of live S strain. So did he inject the live S strain? No, he did not inject the live S strain. Whatever that S strain he has taken, they are all dead. Okay, so he killed them, he ensured that all are dead, and then he mixed, okay, with the live R strain, then injected into mice, and to his surprise, okay, actually the mice should not die, because here the he already killed the S strain that actually causes disease. But okay, to his surprise, he saw mice died, and the blood samples from those dead mice have shown the presence of live S strain. Okay, so this, what is the reason for this? Griffith was not able to explain. Okay, and concluded that R strain transformed into S strain. Okay, so that's what is called transforming principle. So we come to know that Frederick Griffith discovered transformation in bacteria, right? So he concluded finally that R strain somehow transformed into S strain. But what is that transformed the R strain into S strain? We know in case of these bacteria, they will be having proteins, they're composed of proteins, they're composed of carbohydrates, they're composed of lipids, and are also composed of nucleic acids. Now, out of all these, right, which one is responsible for transformation of R strain into S strain? That he could not explain here. Okay, so the R strain transformed into S strain. So that is what the R strain developed. Okay, polysaccharide coat, that is mucus coat, and it became S strain. So but what is transferred from this S strain to R strain and made the R strain to become, okay, S strain, became a question mark, and Frederick Griffith was unable to explain. And even though he was unable to explain, okay, so this phenomenon, so later some other scientists continued the experiments and they came to a conclusion. So what is that conclusion and all, that we will see in the next session.